I am honored to present tethered cord syndrome in children at the Grand Rounds. My talk is to describe the pathophysiology of tethered cord syndrome related to its diagnosis and treatment. The concept of spinal cord tethering or traction originated in the 1950s and 60s. Several articles suggested that traction of the spinal cord by caudal anomalies can be a cause of reversible neurological deficits. Despite logical assessment of their patients, the concept was met by skepticism. In 1976, Hoffman et al. In Toronto, Canada, selected 31 unique cases for study. The patients showed motor and sensory deficit in lower limbs and incontinence associated with an elongated cord and thick phylum terminale. After sectioning the phylum, relaxation of the spinal cord was noted, and neurological improvement followed in these cases. Hoffman coined the term tethered spinal cord to designate combination of the specific anomaly and the symptoms localized in the lumbosacral cord. Next slide. They supported the previous author's concept. The skepticism continued. Many neurosurgeons claimed that the concept was derived merely from visual impression on anatomical abnormalities. We at Loma Linda addressed the skepticism and researched the physiological mechanism of reversible neurological deficit. In 1981, we demonstrated impaired oxidative metabolism in the spinal cord of the patient who presented with the same symptoms and anomaly as those in the Toronto group. Further, we confirmed parallel metabolic and neurological improvement after untethering procedures. The research appeared in the Journal of Neurosurgery entitled Pathophysiology of Tethered Cord Syndrome. Next slide. Immediately, Macron and colleagues accepted our work as scientific evidence for tethered cord syndrome as stretch-induced functional reversible disorder. Additionally, we extended the mechanical cause of cord tethering to lipomas, lipomyelonmeningoceles, and others, and adopted the term tethered cord syndrome. Since the publication of adult tethered cord syndrome by Pang and Wilberger, the same definition of tethered cord syndrome is applied to all age groups. Since the diagnosis of tethered cord syndrome is based on pathophysiology, I would like to introduce our research methodology. Next slide. We combine the three methods, anatomical, biological, and physiological, on cat spinal cords. Anatomically, lumbosacral cord and phylum are divided in 10 equal length segments and stretched by traction. In the graph, the abscissa is for the traction force and or the ordinate for elongation rate. The phylum elongates linearly, much greater than any cord segments. Apparently, the phylum has the highest viscoelasticity. Next slide. Then, what will happen if the phylum loses its elasticity? Traction is applied directly to the cord to simulate human tethered cord syndrome. Here, the spinal cord stretching is remarkable without the buffering effect of the phylum. The greater the traction force is, the greater the elongation. And the lower the cord segment, the greater the elongation. Off traction, the spinal cord length returns to normal. From these results, we concluded that low lumbar cord are vulnerable to traction. This corresponds to the common neurological deficit in the distal legs in incontinence. Now to link anatomical conclusion to the physiological analogy, we studied oxidative metabolism. Now to link the anatomical conclusion to physiological analogy, we studied oxidative metabolism. Slide. The biological principle of this study is that the central nervous system entirely relies on the oxidative metabolism for neuronal activities and cell survival. Mitochondria, the energy factory, ATP, is the energy donating molecule to maintain ion pump. And cytochrome A3 embedded to the mitochondrial cristae is the key enzyme for oxidation. Next slide. ATP is produced by ADP phosphorylation, which is tightly coupled with electron transport. When cytochrome A3 is oxidized, it accelerates electron transport and enhances ATP production. When reduced, it decelerates the transport and slows ATP production. Slide. The dual wavelength reflectance spectrophotometer is the ideal technology to show redox changes of the cytochrome. Two beams of light are projected to the spinal cord. The reflected light from the cytochrome is collected through the microscope into the photomultiplier, and the redox is calculated. In this system, greater the reflection is, the more reduced the cytochrome. In this study, we found that the reduced state was found in both the experimental and human tethered spinal cord, and was similar to those found in hypoxia or ischemia of the spinal cord. To support the biochemical changes, we studied spinal cord evoked potentials and found diminished interneuron potentials in the stretched spinal cord. Next slide. Spinal cord blood flow decreased during traction and returned to normal after its release. Next slide. Summarizing the results, one, there was a parallel between the spinal cord elongation, metabolic impairment, and interneuron deterioration. Two, immediate recovery from these changes occurred after release of cord traction. Next slide. Now the pathophysiology is clarified, and tethered cord syndrome is defined as stretch-induced functional disorder of the spinal cord with its caudal end anchored by an inelastic structure. On reviewing an article on tethered cord syndrome, however, the surgical outcome